Let's look at blood typing. Blood typing is a concept that when you read it in the book, in any book, it's really complicated. And it reads a lot like stereo instructions. And I don't know why, because it's not that difficult to understand, especially if you understand the concept of antigens and antibodies, which we've already discussed, right? Remember, the antigens are the things that are found on the cell membrane, little things that are sticking out that our body uses to identify whether or not it belongs there. The antibodies is what our body makes in response to foreign antigens. It sees something that's wrong, yes, the Y-shaped antibody. It sees something that doesn't belong there, and it makes the antibodies that attack and snap on to those antigens perfectly, like a key in a lock. So if you can understand that concept, well then understanding the blood cell typing isn't that difficult. But for some reason, I don't know what it is, they just have never made a very easy way to understand this. I will make it very easy. If you want to determine blood type, first thing you do is you draw 4 red blood cells. And James, I don't know if they can see this. Can you see if this is visible? Okay, thank you. All right. We have 4 red blood cells because there's basically 4 types of blood. So red blood cells are a little different because they have their own unique set of antigens, their own unique set of things that are sticking off of them. When they were first discovered, they were arbitrarily just given letters of the alphabet, A and B. And it was discovered that a person has these antigens on their red blood cell, and one group they designated group A, and another group they designated group B. Later on, they found that some people have both A's and B's. That came later. But initially, they found out that people had either group A, these type of antigens, or this type, which they called group B. Then they found that some people didn't have either of them. So, since we're going with letters, they were going to make a 0, but since they're going with letters, the next letter would be O, meaning no antigens on the surface of this red blood cell. Not too difficult so far, right? They looked at these red blood cells, they found out that they have one of 2 types of antigens, and they just arbitrarily named them. Well, this time, this one we'll call A, so everybody who has these types, we'll just say they have A. And everybody who has this type of antigens, we'll say they have B. And if they have no antigens, we say they have 0 or O. And then later they found out that some people actually have both, A and B. All right, now, what the body does in this case, if a person has A antigens, the body automatically makes antibodies for what it doesn't have, which is what? B. So if a person has this type of red blood cell with A group antigens, the body automatically makes antibodies against what the person does not have, which is B. So if they have A antigens, they have antibodies for B, which means if we put this red blood cell into this guy's body, these antibodies attack it, they stick together, and they clump, which means all of those nice red blood cells that are supposed to be single and be able to move freely and flexible end up as one big clump of red blood cells, which is not going to be very beneficial to the patient, right? Bad news. So, this guy, we found out, he can only get type A. We can't put this guy in his body. Now, this guy doesn't have any antigens. So we found out that we can put this red blood cell into this guy's body because there are no antigens to be attacked for the things to connect to. So this guy can get group A or O. If we put this red blood cell into this guy's body, these are going to attack, right? Not completely, but they'll attack here and here and here and here, which means they'll be clumped together, which means it won't work. And this is bad. So a person with group A antigens on their red blood cell can only get group A blood or type O. No antigens, type O. So if we look at this guy with B's, group B blood, his body's automatically going to make antibodies against what he does not have, which in this case is A. Which means he can only get B, or he can get O. Can he get A? No. But what about this one? No. Right? Because the antibodies are going to attack. You see how that works? All right. So not so bad so far, right? Okay. Now, this guy here, AB. Let's figure for a minute. We have group A. His body's automatically going to make antibodies against what he doesn't have, which are B's. This guy here with B antigens, his body's automatically going to make antibodies against what he doesn't have, group A. This guy here has both A's and B's. So is he going to make is he going to make antibodies against A? No, because he would attack himself, right? That's not going to work. Is he going to make antibodies against B? No, because he would attack himself, right? So is he going to make any antibodies at all? Not against red blood cells. So far. You see how that works? So, can we put group A blood, red blood cell into this guy's body?
Yes.
How many say yes? Because does he have any antibodies? No. So this can go into this guy's body without a problem. What about B's?
Yes.
Yes. What about ABs?
Yes.
Of course, it's his own type. What about O's?
Yes. Sure.
Yes. This is why he is called the universal receptor. He can get any type, recipient, excuse me. He can get any type of blood cell. He is the universal recipient. He can get A's, he can get B's, he can get O's, he can get ABs. Now, we come to type O. His body is automatically going to make antibodies against what he doesn't have. What doesn't he have? Well, he doesn't have A's. And he doesn't have B's. So even though this red blood cell, the blood cell can go into this guy, right? And he can go into this guy, and he can go into this guy, and he can go into himself, which makes him the universal donor. Can he get this? Because he has antibodies that will attack it. Can he get this? Because he has antibodies that will attack it. Can he get this? Because he has antibodies that will attack it. He can only get himself. But he can give to anybody. So I call him the universal donor. Make sense? A little bit easier than how this stuff reads in the book, right? Trust me, it is. Okay. Now, somebody is going to ask, well, wait a minute, because you're leaving something out. Because I was told that I'm A positive. So what's the positive mean? That's a good question. Okay, this worked out really well for a while. And way back in week number 1, we were talking about the differences in antigens. I said that the bigger the difference, the bigger the attack. If it's a really big difference, like an A to a B, huge attack. The body recognizes it's as it being very, very, very different and attacks with great force. If it's a tiny difference, there's a tiny attack. Which is why, when they first discovered this one, and then having them 4, it worked pretty well for a while. The results were pretty good, but they weren't perfect. The reason for that is because they later found out that there's another group of antigens. They're small. That's why they were missed, because the response to them wasn't really great. But there's another group of antigens. They were discovered in testing the rhesus monkeys. Those were the test subjects. Not like Reese's Pieces, rhesus monkeys, RH. So they called this antigen, when they found it, the rhesus factor. And they found that people either have the rhesus factor or they do not have the rhesus factor, which means they're either rhesus factor positive or rhesus factor negative. So what we would do, if we found this person's blood had A's on it, A antigens, and we found they had a rhesus factor, we would say that this person has type A blood, rhesus factor positive. Type B blood, rhesus factor negative. Now, because we like to abbreviate things, right, we decided to just say RH for rhesus, the monkey, and a negative sign. So we would say this person here is type B RH negative. Then, that wasn't short enough for us, so we just say that they are B negative. A positive. Does that make sense? Okay. Now, let me read through this little part right here very quickly on page 83 of the note packet, the only part that I've bolded here, but just for classroom discussion. Type O blood can be given to anyone with any blood type. This is why people with type O blood are called universal blood donors. AB positive is universal recipient. Because again, if they have this, the positive, they can get either positive or negative. If they are negative, they have to get negative. If they are positive, they can take positive or negative. If they are negative, they have to get negative. Because remember, if they're positive, they're going to create antibodies against not having them. They can't make antibodies against this because it'll attack themselves. So there won't be any antibodies against the rhesus factor. If they're negative, they'll have antibodies against the rhesus factor. So if a person is positive blood, they can get either positive or negative blood, whatever the type might be. If they're negative blood, they have to get negative. Blood typing is especially important during pregnancy. If the mother is found to be Rh negative, the father has to be tested. If the father has Rh positive blood, the mother needs to receive a treatment to help prevent development of antibodies that can be dangerous to the baby. It's called Rh incompatibility. If you're Rh positive, you can receive Rh positive or Rh negative blood. If you're Rh negative, you can only receive Rh negative blood. Now, about 85% of the population is positive. So, and then if dad is positive and mom is negative, there's about a 50% chance that the baby's going to be, that there's going to be a problem. Now, this is only going to affect not the first child, but the children after that. Because the initial child won't be affected, so there won't be enough time for mom and body to make the antibodies. And during the pregnancy, it's not really a problem. It's only at the end of the pregnancy when the delivery is occurring when there can be a mixture of blood. So, if it ever comes time for you to have to figure out blood typing, rather than memorize who can get what, because that is going to confuse you, because it confuses everyone. Rather than memorizing it, get a blank sheet of paper and draw 4 circles. Put A's, B's, AB's, and O's. Then you know A's have antibodies against what it doesn't have, the B's. And then do that for all of them. First you determine the type, whether or not this person can get A's or B's or AB's or O's. That's the first part. Then you go back and you do the RH part. If they're positive, positive, they can get either positive or negative. If they're negative, they have to get negative. But break it down into those two parts, and it makes it much easier to figure this out.